So, yeah, good afternoon, everyone. So thank you very much for the invitation to talk at this workshop. As the title of my presentation suggests, I will be talking about dengue in Queensland and, of course, the network science that was involved in the work. So I'll begin by introducing CSIRO's Disease Networks and Mobility Project. So this is a project that I've been working on since I joined CSIRO in 2017. And the work that I will be presenting today is also part of this project. Then I will tell you a little bit about dengue in general, how it is spread, and a bit of, epidemiology about dengue, how it works. And then I'll move on to present the work that we have been doing around improving surveillance in Queensland. So the Disease Networks and Mobility Project, or DiNeMo for short, is an interdisciplinary research project that aims to understand how human infectious diseases might arrive and spread in Australia. So what we do is we analyze big data sets and we try to identify patterns of people movement, both internationally but also domestically, to forecast the spread of different diseases. So mainly our work when we started was focused on dengue, and that's what my talk is about today. But then obviously with the start of COVID, we kind of moved into that area and doing work there as well. So just to summarize, like what we are hoping to do with this work is to contribute to Australia's preparedness for future epidemics and pandemics. Now, a major aspect of the project is human mobility and behavior, which plays a major role in disease spread. And I guess I really want to emphasize here that, say, if you think about dengue, for example, I guess most people straight away think, yes, it's a mosquito-borne disease, so it's spread by mosquitoes. And that's definitely true. But what a lot of people don't realize is that most of the spatial spread is actually due to human movement, because the mosquitoes are largely stationary, they stay in one place, and it's people who move around and carry it to different places. So this is one of the main focuses of the project. All right, so dengue. As I said, I want to tell you a little bit about how it is spread and a few important things about dengue. So I'm not sure how much you already know about this virus, but in my experience, generally, I feel that awareness around dengue is not very high. And that definitely includes myself as well. Before I started working on the project, it wasn't really on my radar. I didn't know much about it. And I definitely didn't know how serious the spread of dengue is. So as I said, it's a mosquito-borne disease. But what many people don't realize is that the WHO pronounced dengue the most important mosquito-borne disease. And it is because it's estimated that there are 50 to 100 million infections each year globally. And more than 100 countries are now endemic. So that means that the disease is in constant circulation in the population, and approximately half of the world's population is at risk of contracting the virus. So it's definitely important research. It'll be beneficial to a lot of people. In non-endemic countries like Australia, so Australia is non-endemic, that means the disease is not in constant circulation, but we've got the vector in North Queensland. And here it only takes one imported case to trigger an outbreak, a local outbreak. So you might remember in 2009, there was a big outbreak in the Cairns region with more than 900 infected people in less than 8 months. And researchers later found that that outbreak was triggered by a single person who got infected in Indonesia, then came back to Australia, was still infectious on arrival, and triggered that whole outbreak. So it's really just that one person that is needed to trigger an outbreak like that. Now, in terms of preventing dengue in Australia, Queensland Health has put together a dengue management plan for efficient surveillance and control of the disease. And vector elimination is the first line of defense for Queensland Health to prevent the wider spread of dengue. Now, when a dengue case is notified, it is referred to an entomologist. And the reported case, if the reported case is in a region where we know that there are vectors present, then there are several control measures like indoor residual spraying, and people go around and empty out the water containers where the mosquitoes might breed in. So the aim of this is to eradicate the virus by eliminating the vector. So Queensland Health is basically trying to kill off the vector before it can spread the virus to other people. Now, this kind of control is reactive, right? So Queensland Health gets notified, and only then they send out their personnel to carry out the spraying. And this is effective only when this is conducted close in time to the infectious period of the notified case. So theoretically, if we manage to get rid of all the mosquitoes before the end of the extrinsic incubation period, so that is before they get infectious, the mosquitoes, then this kind of control works really well. But prompt reporting of cases is not always possible. There's various factors involved. There's awareness, willingness of patients to go to the doctor. People might not even know that they have dengue because symptoms are flu-like, so they might not be very severe. But then also training and diagnostic abilities of doctors. They might not always recognize that it's dengue. They might think it is just a cold or the flu. So yeah, there's various factors involved, and cases are therefore not always promptly reported, and sometimes they're not reported at all. So the next slide here shows you a diagram that exemplifies dengue transmission from one person to another. So as I said, in Australia, outbreaks are triggered by imported cases. So say Alice arrives infectious in Australia, so she caught the virus overseas somewhere and is infectious on arrival. She then might not feel sick and goes about her usual business and lives in an area that has mosquitoes, dengue mosquitoes. And now Alice is bitten by a mosquito. Now this mosquito then gets the virus. And that mosquito undergoes an extrinsic incubation period. So that's this bit here. So during that period, the mosquito is not infectious, but it has the virus already. And then at the end of the intrinsic incubation period, it is infectious and can spread the virus to other people. So the mosquito stays infectious until it dies. And during that period, it might bite other people. So if Bob gets bitten by that mosquito, then Bob undergoes an incubation period as well. And after that period, Bob gets infected and then could get bitten by other mosquitoes who then get the virus, and so on. Okay, and now I'll actually come to the work. So I talked a lot about the DiNeMo project and how dengue actually spreads. But yeah, I'll now come to the actual work, which is a collaboration between members of the DiNeMo team, QUT and Queensland Health. So this work is currently under review at +1, and a preprint is available on medRxiv if you're interested. So the main question that we were trying to answer is how we can minimize the risk of future dengue outbreaks by improving this whole surveillance system. Now, previous studies, they've mainly looked at timeliness and of disease surveillance, and they measured it by comparing average delays in notification to some predefined standard. But what we do differently is we quantify the efficiency of Queensland's surveillance system by actually measuring the number of secondary cases that were caused, and we also look at the total outbreak size. And to be able to do that, we actually have to construct a network. So to construct this network, we use dengue case data that was provided by Queensland Health. We have data covering the period from October 2001 till September 2018, with about 5,200 cases being reported in that period. So for each reported case, we know the date of symptom onset, the date of specimen collection, date of diagnosis, and also date of notification. We also know the place of residence, which is recorded as at a suburb level. We also know the place of acquisition, which is if it is in Australia, it's recorded at postcode level, and if it's overseas acquired, then we just know the country. And we also know the dengue strain. So as I said, from this data, we construct a network of dengue cases. And edges in this network represent possible disease transmission paths. So for a case I to have triggered a case J, there are several conditions that have to be satisfied. So first of all, both cases have to have the same strain. Second, the place of acquisition of case J must coincide with the place of residence of case I. And this is basically, there are some assumptions here, but this is basically all we can do with the data that we have. But it is a reasonable assumption because mosquitoes are stationary. So they move around 200 meters or something. So they don't travel large distances. And then to also respect the temporal epidemiology of dengue, the onset date of case I has to be greater than the onset date of case J minus the sum of the intrinsic incubation periods, that's the incubation period in humans, the infectious period in humans, the incubation period in mosquitoes, and the infectious period in mosquitoes. And lastly, the onset date of I has also to be less than the onset of J minus the sum of the incubation periods in both mosquitoes and humans. So this is a mouthful, and so I'll just go back to this diagram to kind of exemplify that, because it would make more sense if I show it on the timeline here. So basically here we've got this transmission, the transmission between Alice and Bob, right? But so say we've got a case who got infectious somewhere here within the mosquitoes EIP, then there's no way that that case was directly linked or triggered by Alice, because that mosquito was not infectious at that time, so it couldn't have triggered that case. And then again, if say Bob became infectious somewhere here after the mosquito already died, again, there's no way that that case was directly linked to Alice. Maybe there was a different case in between and we can trace it back to Alice, but there would not be a direct link between the two. So we use all these conditions to create this network of cases, and that's going to look something like that. I'm sorry about the resolution, it's not so good. But what you see here, the nodes, they're dengue cases, and this is just like a small part of the bigger network. I didn't want to show the whole thing because you wouldn't be able to see anything. But the cases here, that dengue cases, sorry, the nodes are dengue cases, and then the edges, they're directed and they show possible transmission paths. Now you can see here that a lot of the nodes here, so this one, for example, has a lot of incoming edges. And in reality, every node only has one parent, right? So what we want to get is a direct acyclic graph. So we need to prune this network. We want to get rid of some of these edges that are not very likely. And to do that, we use the latent influence point process model. I'm not going to go into too much detail here because that's not really part of this work. So this model was also developed by the DiNeMo team. And if you're interested in the details, there's reference at the bottom of the slide that you can look at. But just in short, the LIB model is based on a multivariate Hawkes process. And in our case, it estimates pairwise causation probabilities between dengue cases. So the model considers environmental heterogeneity, so vector presence, for example, mobility between regions, and also the decay in infectiousness. And by doing that, it associates a probability with each edge in the network. And nodes, so nodes that have more than one incoming edge what we do is we delete the edges with the lowest probabilities and only keep the one with the highest one, because that's the most likely transmission path. And then what we get is this directed acyclic graph, where every node just has one incoming edge, but say this node here, one node can trigger a lot of cases, but each case only has one incoming edge. And then here, for example, these nodes here, the blue ones, they're imported cases, so they don't have any incoming edges because we don't actually know what's been going on overseas. That's not what we're interested in. We just know that they were imported into Australia and then they triggered local cases. Okay. So how do we actually reduce this or minimize the outbreak risk? So before I get to the actual network analysis that we've done, I want to briefly talk about the different delays that we can see or identify from the dengue data. So there's 3 delays in total. First, there's the delay on the side of the patient, which is the time between the date of symptom onsets and the date of specimen collection. So a person gets sick, maybe waits a couple of days, then goes to the doctor, and then the doctor orders a dengue test. So there's a bit of the delay there. Some people might go straight away, but others might just wait a few days and see how they go, and then they decide to go later if symptoms get worse. The second delay is the laboratory delay, and that's the time from the specimen collection to the date of diagnosis. So that's so the test needs to be evaluated, and then we know from Queensland Health there's often delays because labs get swamped. They're obviously not just dealing with dengue; they're also dealing with other diseases that might have higher priority, and dengue tests might sit there for a while until they actually get processed. And then lastly, there's the clerical delay, and that's the time between the diagnosis and Queensland Health being notified. So tests are conducted and they might come back positive, but again, maybe labs are too busy and they don't notify Queensland Health straight away. It might take a couple of days. So ideally, we want to minimize these delays to be, for surveillance and response measures to be as effective as possible. So if you look at the figure here at panel A, it shows that the lab and clerical delays gradually declined throughout the study period. So this is great because that actually means that we're learning and getting better at doing the surveillance. So the average lab delay decreased from about 7.5 days to just over 3 days between 2003 and 2018. And the clerical delay fluctuated around 1 to 3 days between 2002 and 2007, and then fell to 0, and then kind of remained constant for about half a day. And I think that's great. That at least once we know that someone is infected, that case directly on the same day gets notified to Queensland Health, so they can send out their personnel. Overall, we noticed that variation in the overall notification delay from 2008 onwards were largely influenced by lab and patient delays. As I said, the clerical delays were pretty constant at half a day. And then we also looked at the spatial effects, so that's panel B. And we can see that Townsville saw the longest clerical delays on average across all the regions, and it was closely followed by Cairns. So Townsville and Cairns, that's the 2 main regions where cases are reported. That's where most of the large outbreaks happened in history. Now, an average lab delay of at least 4 days was reported for most of the other regions. And on average, the patient delay was about 4 days, which is not too bad. Could be better, but it depends. So as actually at this point, I should say as well, I forgot to mention that earlier when I showed you that diagram, the intrinsic incubation period in mosquitoes varies depending on environment. So that's temperature and humidity. They're the 2 main factors that influence the intrinsic incubation period. So if weather conditions are ideal, it might be as short as 2 days. And if that's the case, then Queensland Health really needs to act quickly. During the winter months when conditions are not ideal, the period is longer. It might actually be longer than the mosquito lives, and so that's why we don't see that many outbreaks during winter. But now getting back to the actual work. Sorry, slides are stuck. Can you see the slide with the transmission period? Sorry, just double checking because my slides got stuck for a bit. Okay, perfect. Okay, that was one too many, sorry. Okay, now. Okay, so transmission period of cases. So the transmission period of a case begins at the end of the mosquito's incubation period and persists until the death of the mosquito. So of course, a person can be bitten by more than one mosquito. And therefore we define the transmission period for a particular case from the start of the earliest concluding EIP till the end of the death of the last mosquito. So overall, the network analysis showed that approximately 10% of all reported cases resulted in secondary infections. And we call these cases fertile cases. And this is what the figure here shows. That's the distribution of transmission periods for these fertile cases. So if we look at the chart for Cairns, you can see that half the fertile cases had a transmission period of at least 7 days. So again, that's quite long if you want to prevent outbreaks. And this period can be shortened by shortening the 3 types of delays that we talked about earlier. So remember, as soon as Queensland Health gets notified of a case, they send out this personnel to spray the areas and kill the mosquitoes. And hence, we want to shorten all these delays so that Queensland Health gets notified quicker and then they can send out their personnel quicker. Hopefully they manage then to eradicate the mosquitoes in time so that they can't infect any other people. So to minimize the outbreak risk, we tested various thresholds of response delay. And this is what's shown in these figures here. So what we did is we enforced thresholds on the network that we constructed earlier by removing the child cases and their descendants. So for example, if we say, or if we enforce a response period of 5 days, then we look at what cases could have been prevented during those 5 days. Which cases would have not occurred if we had killed off the mosquitoes during those 5 days. And then those cases were labeled as prevented cases, and that's how we measure the effectiveness of surveillance. So the highest reduction was achieved for the response delay threshold of 5 days. So it's not surprisingly, the shorter the better. But we also noticed that there was a dramatic drop in the percentage reduction if the threshold was increased to 10 days. So for example, so we could have managed to prevent about 87% of dengue cases if this 5-day response threshold was enforced. But if we then increase the threshold to 10 days, we can only prevent about 40% of cases. So we noticed something similar for outbreak size. And that's, again, they're closely related. It's really just another measure, right? So outbreak sizes were reduced by about 80% if the threshold was at 5 days, but then also dropped to 40% if we make this response delay longer, if we move to 10 days. In summary, we constructed this disease spread network where edges represent the most likely disease transmission path. And that enabled us to test the efficiency of the current surveillance system and also to look how we can best improve it. So we found that, and again, this is not very surprising, we knew that already, that short, like the quicker you respond, the better the outcome. But I guess what was surprising is that if you increase your response to live from 5 to 10 days, there's a huge drop in the cases that could be prevented. So we talked about the 3 different types of delay. And I guess the delay in patients, it's a little bit harder to tackle because ultimately it's up to the person if they want to go and see a doctor or how quickly they go. And as I said, sometimes symptoms are really mild and you might not realize that you have dengue. But I guess one thing that can be done there are obviously awareness campaigns, and Queensland Health is already doing that. But what our analysis does is that through this analysis, those awareness campaigns can be more targeted, say in regions where there's more occurrence of dengue or at certain times of the year. But then the other delay, which is on the side of the doctors who are treating the patient, that's actually where we can really do something, because once a person goes to the doctor and the doctor suspects that it is dengue, we can then urge the doctors to kind of process that quicker, order the tests, maybe already notify Queensland Health. Some doctors are already doing that. So that's, again, we know that from communication with Queensland Health, some GPs are already notifying it straight away. So they don't wait for the outcome of the test. They just say, we've got a probable case here. And Queensland Health is really appreciative of that because they already send their personnel out anyway, because they do have that capacity. And I guess this is that's kind of a point in the system where we can really try to shorten this delay and be more efficient. But I guess ultimately, or the next thing that we would like to do is rank incoming tests. So once the doctor has ordered the test, it needs to be processed by a lab. And we would like to then prioritize, because currently it's first come, first serve. And that might not be ideal, because you might have a test there that was ordered quite late. So that person may already be infectious for a while and has the potential to trigger a large outbreak. So ideally you would like to process those cases first to have a definite answer there, so that case can be quarantined and then you can do all your other control measures. Yes, and that's it.